Hello everyone, I'm Chen Jiayi from Northwestern Polytechnical University. In this video, I will introduce our work, Think Twice Before Selection, Federated Evidential Active Learning for Medical Image Analysis with Domain Shifts. Federated learning enables collaborative learning across multiple clinical institutions to attain a powerful global model through model aggregation, while preserving local data privacy. Unfortunately, such a learning pipeline requires each client to prepare its own labeled data, whose scale is constrained by the available expertise, time, and budget for data annotation. One possible solution to reduce annotation costs is leveraging federated active learning to identify and annotate a subset of informative data for each client. However, domain shifts across clients, evidenced by low p-values in kernel density estimation of energy scores, pose challenges of overconfidence and limited uncertainty representation for federated active learning. Specifically, existing methods evaluate data uncertainty based on the softmax prediction, which is essentially a point estimate and can be easily miscalibrated on data with domain shifts, resulting in unreliable uncertainty evaluation. Besides, uncertainty can be divided into aleatoric uncertainty and epistemic uncertainty. The former reflects the inherent data complexity, where the latter captures the restricted knowledge of a model caused by insufficient data or domain shifts. The softmax prediction can represent aleatoric uncertainty but fails to capture epistemic uncertainty, leading to incomplete evaluations. To deal with the aforementioned challenges, we proposed a novel method called Federated Evidential Active Learning. It comprises two iterative phases, federated model training and local data annotation, where federated model training involves model distribution, local training, and model aggregation. First, I will briefly introduce the Dirichlet-based evidential model utilized in our study. The classical convolutional neural networks utilize the softmax operator to transform the logits into the prediction of class probabilities. However, this approach essentially provides a single point estimate and can be easily miscalibrated on local data from diverse domains. By contrast, the Dirichlet-based evidential model views the categorical prediction as a random variable following a Dirichlet distribution. Given a sample and a model, the probability density function of categorical prediction can be derived, where the Dirichlet parameter is linked with evidence and can be calculated based on the output logits. In calibrated evidential sampling, we integrated both uncertainty and diversity measures to identify the most informative samples for annotation. As for uncertainty evaluation, we use the expected entropy of all possible predictions to deliver the aleatoric uncertainty to measure the inherent complexity present in local data. In addition, we employ the differential entropy of the Dirichlet distribution to quantify the epistemic uncertainty linked to domain shifts between the global model and local data. The overall calibrated uncertainty is evaluated by considering aleatoric uncertainty in both global and local models and epistemic uncertainty from the global model. Besides, we also adopt local constraints to ensure diversity among selected samples. In evidential model learning, we adopt the base risk of cross-entropy loss as the task loss for classification, to consider all possible predictions and elevate overconfidence. We also incorporate evidence regularization to further reduce incorrect evidence and improve correct evidence. We evaluated the proposed method FIL on 5 real multicenter medical image datasets, comprising 2 classification and 3 segmentation datasets. The details of datasets, evaluation metrics, and backbones are provided as follows. We compared FIL with 8 SOTA methods. The first 6 strategies are developed for standard active learning, while LOGO and CAFL are specifically tailored for decentralized scenarios. The experimental results indicate that FIL achieved superior results on both Fed-ISIC and Fed-CAMLON datasets. Additionally, it is noteworthy that FIL surpasses the fully supervised performance after the 3rd round of active learning on Fed-ISIC. As can be seen, FIL also exhibits superior performance on 3 multicenter segmentation datasets, as evidenced by its higher dice scores. We conducted ablation studies to analyze the effectiveness of each component. We also visualized aleatory uncertainty on Fed-PolyP dataset. It is noticeable that global aleatory uncertainty and local aleatory uncertainty highlight different regions, underscoring the importance of combining aleatory uncertainty in both models for comprehensive assessment. We further analyzed the Dirichlet simplex on a subset of Fed-ISIC dataset, encompassing 3 classes. When selecting samples with FIL, the Dirichlet distribution becomes more concentrated at the simplex corner for unlabeled samples, indicating reduced epistemic uncertainty in the global model. This trend verifies the effectiveness of calibrated evidential sampling in addressing domain shifts. Additionally, starting with an identical set of label samples, we tracked the selection of samples in the second Federated Active Learning round, utilizing various methods. The Dirichlet distribution of samples selected by FIL showcases a broader spread across the simplex, indicating that FIL effectively models the global model's knowledge of local data and identifies samples with high epistemic uncertainty. In summary, there are 3 main contributions in our work. Firstly, we explored a rarely studied problem, Federated Active Learning with domain shifts. Secondly, we proposed a novel method, Federated Evidential Active Learning, with a sampling strategy and a local training scheme to tackle the challenges in Federated Active Learning with domain shifts. Thirdly, we conducted extensive experiments and analyses on 5 real multicenter medical image datasets to verify the effectiveness and superiority of our FIL method. That's the overview of our work. We appreciate your time and interest in our research. Thank you for watching.